Hi everyone, I'm Omar, and we're here at the Bay Area Science Fair. This is over 400 students and over 260 projects we're about to see inside this fair. So let's go in and check it out. So we're here today with,
Kamila Machalski,
and how old are you?
16.
And you want to tell us a bit about your project today?
Yeah, so I did it on Alzheimer's disease, which is a progressive neurodegenerative disorder that results in an irreversible loss of neurons in the brain. I looked specifically at brain-derived neurotrophic factor, or BDNF, which is a neurotrophin found in the brain responsible for the repair of already existing neurons. So in the human cortex, decreases of these expression levels have been reported, and as for the hippocampus, increases and decreases have been reported. So I wanted to look at the expression levels of that in the human hippocampus. So basically, I had a whole process. I had to extract the RNA, reverse transcribe it into complement DNA, and I had to amplify it so that I was able to quantify how much of this gene was expressed in each sample. I had 20 samples in total. 10 were Alzheimer's disease and 10 were control, frozen hippocampal brain tissue. So in the end, just an overview, the total BDNF expression level in Alzheimer's disease subject was 3 times lower than that of control. So I did find a significant difference in that.
So I noticed you're doing got some cDNA and some RT-PCR. So how do you get how did you run these experiments? Where did you get your hands on some of these?
I started volunteering at Margaret Fanistock's lab in the summer, and they actually specialize in Alzheimer's disease experiments, I guess, and that's how I started there. So they had all the equipment there.
So pretty impressive project. You can see a lot of work went into it, and it's fairly high level. This is like university level, at least second, third year. So yeah, it's pretty impressive. So what got you into Alzheimer's? Just because you worked in the lab, or was that in the field for you?
I worked at the lab, and it was just always, like, my grandma has Alzheimer's. It was just always around me, I guess. So that's how I started there.
So you can see this is pretty advanced stuff. Thanks a lot for your time. I wish you all the best in your project.